It's a pleasure to talk today to a little bit about our work, and I'm going to talk about two subjects. First, some work we've been doing with a collaborator on Alzheimer's disease, looking at the effects of diet, and then if there's time, a broader topic of looking at biomarkers for personalized health that we've been involved in for some time now. Let me first start by thanking the people who are doing the work. So Angela Hansen is our collaborator, who's an expert in Alzheimer's disease and got interested in metabolomics a few years ago. And then the people who did the work in our lab, primarily Lisa Betcher, did the lipidomics, and Daniel Djukovic, who did some aqueous profiling, and Nagana Gowda is our long-term expert in NMR. And then collaborators in nutritional biomarkers are Joanna Lampe, Ross Prentice, and Cheng Zhang did work at primarily the Fred Hutch. And the work is done through our metabolomics center called the Northwest Metabolomics Research Center. You can see the website there, and the work is primarily supported by the NIH. We are very interested in both broadening the measure of the metabolome and improving its measure through improved data quality. And I show this figure to those of you who want to see, sort of the scope of where the current status is in terms of measuring different parts of the metabolome. And in general, we want to measure as many metabolites as we can, but the limitations of being more quantitative, more accurate, and reproducible often require us to measure, fewer metabolites. So we're doing untargeted global profiling. We may be able to measure 1,000 or 2,000 identifiable metabolites nowadays, but if we wanted to improve our control over the metabolites that we measure and get improved performance from those assays, we often move into a targeted mode. And we've done quite a bit of work in developing those types of assays. And then if you want to be, to absolute quantitation, that tends to be even a smaller number. Now, in terms of lipidomics is a little bit of a special case, as I show here at the bottom, because the similarity of lipids across classes allows one to be actually quite quantitative with a relatively modest number of internal standards. And I want to underline there's no universal detector for metabolomics, so we're really just looking at a small portion of the metabolome in any case. So one of the interesting observations in the area of Alzheimer's disease is that eating a Western or high-fat diet is a risk factor for developing Alzheimer's disease over time. And there's not a lot known about the mechanisms. However, if you're an APOE E4 carrier, that you have a gene for APOE E4 lipid carrier, then you actually can benefit from high-fat feeding, which is really interesting and sort of contrary to the overall risk profile. So we'd like to investigate this more. We'd like to know, what are the lipids and free fatty acids that cross the blood-brain barrier and enter the brain. We know that the brain is not able to synthesize these, and so it must transport them in. We'd like to know what the fate is of the free fatty acids. Now, it's not very easy to investigate these in live patients, obviously, so we have to make some compromises. And so we're going to be looking at CSF instead as a proxy for what's going on in the brain. Angela Hansen did some work a few years ago that showed that older adults who were given either a high-fat or low-fat meal, if they were an E4 carrier, actually showed quite a big increase in their cognition scores after a high-fat diet. And so this prompted ultimately a metabolomic story to start investigation to try to understand what's going on. We know that the APOE E4 becomes lipated. It becomes a better at targeting A-beta proteins for clearance, which is one of the components of Alzheimer's disease. And generally, it needs a higher concentration of lipids in order for it to become active. And it's also somewhat less stable than the more common form E3. So it's an interesting category of patients. And of course, APOE E4 carriers are at higher risk for developing Alzheimer's disease. So the goal here is to try to take a first look at the effects of a high-fat diet on Alzheimer's patients and APOE E4 carriers, and look at the metabolome in the CSF, both the lipidome and aqueous metabolites ultimately. And so there were 21 patients in the study who were given either a high-fat diet or saline, this was by infusion, saline infusion or a triglyceride high-fat infusion. And then their CSF was taken after 5 hours after the infusion. So it was crossover design, so there are 42 total samples. And this is a composition of the TG infusion. You can see primarily oils plus some A components. And the study composition here was 10 women, 11 men. 8 of them were positive, and 9 had cognitive impairment. And they were relatively tight ranges in terms of age and BMI, some range in the triglyceride levels. We perform quantitative lipidomics using the SciEx Metabolon platform that Martin Gira described yesterday. And in this, we typically detect 700 to 900 lipids in blood across 13 different lipid classes. And it's a highly reproducible and quite accurate platform, I would say. And because of the 54 internal standards, you can calculate absolute concentrations of lipids, which is advantageous for a number of reasons, including, comparing across studies. For this study, we extracted the lipids from CSF using dichloromethane and methanol. We used a lipidizer, as I mentioned, and we targeted about 1,100 lipids. We detected a total of 373 different lipids in the CSF. CSF is a kind of a thinner fluid than blood, so it's not too surprising. And depending on each sample, the range was about 160 to 280. And we deleted the lipids that had less than 80% prevalence, and then used k-nearest numbers to fill in the missing values by interpolation. And the CVs were quite good. So certainly, we're right in the range of what we expected for the instrument. So we measured 12 different lipid classes, although there was only one species detected in the lysoceramides. But nonetheless, we saw a significant increase after the triglyceride infusion of the acylglycerols and the lysophosphocholamines. And then we also saw a decrease in the dihydroceramide lipids class. There were no changes in the overall free fatty acid class. However, specific free fatty acids did change depending on the infusion. And so these 3 non-saturated free fatty acids showed some significant changes even after false discovery rate correction. And then in terms of the specific triglycerides, there was not a lot of change, to be honest, based on the infusion. However, overall, but a couple of the components of the triglycerides, so the linoleic and oleic acid containing triglycerides, did increase, and they changed depending on the E4 status, which was pretty interesting. Overall, in the CSF samples, the free fatty acids are the most abundant, and then followed by cholesterol esters. And then, the brain, as I mentioned, does not synthesize free fatty acids. It can synthesize cholesterol. We didn't observe, as I mentioned, a free fatty acid change in terms of as a class, but there were components that did change depending on the diet here, and also the acylglycerols were higher after the high-fat diet infusion, and then there was some triglycerides that changed depending on E4 status, which is interesting. So results are related to some previous work that had shown that the blood-brain barrier transport can depend quite a bit on the specific free fatty acid species. And this is in line with what we saw, which was that there were specific free fatty acids that did change, although the overall probably swamped out in terms of when you compare the entire class of free fatty acids. And probably similar for the triglycerides. There were a few that changed depending on E4 status, but overall, probably the change was swamped by just a large number of triglycerides that we can detect in these types of samples. So we need additional work, I would say, probably in a mouse model to really better understand what the transport is, so we can measure on both the CSF side, the brain, and as well as the blood. Gone on more recently to look at ketone levels to try to understand what is happening in Alzheimer's patients. So it's generally known that the glucose metabolism is low in the brains of Alzheimer's patients. It's also related to E4 status. And we know that the brain can metabolize ketone bodies, but we don't know what is actually driving the levels. For this work, we performed aqueous targeted profiling. So this is a platform that we have built over the years, improving the number of metabolites over time. It's currently targeting a little over 360 aqueous metabolites and uses a SIG 6500 and high-light chromatography, and produces quite reproducible results. Been used in a number of studies. At the time when we were looking at about a little over 200 metabolites from 50 pathways, you can see the reproducibility was good. And we have absolute quantitation for a number of about 32 or 3 metabolites typically. In the samples that we analyzed for the CSF, we were only able to detect 81 metabolites, but they still were detected reproducibly. And to show you that the major changes that we saw, the most interesting actually was 3-hydroxybutyrate, which was...
Daniel, excuse me. We are temporarily using you. Could you speak more louder?
A little louder? Okay.
Yes, or closer to microphone.
Okay. It's right on the side of my face here, so
yeah. So
I'll try to hold it.
Thank you.
So the 3-hydroxybutyrate was the largest, or had the lowest p-value, and had a sizable fold change of increase of 1.7, as well as some other interesting metabolites that were observed to change. And we took a closer look at the 3-hydroxybutyrate. It's reasonably well correlated with a number of other interesting metabolites, including several other ketone body type metabolites, as well as some energy-related species too. And what's particularly interesting about 3-hydroxybutyrate is that it seems to be correlated with cognitive impairment. So patients who have cognitive impairment show a quite a bit higher level of 3-hydroxybutyrate in their CSF, after a high-fat infusion than those who get the saline. And so this is a significant change in the 3-hydroxybutyrate level in the CSF. It might indicate a smaller transport into the brain, which might explain lower ketone body levels in the brain in such patients. So this is some early work, and we're very interested to follow this up with some more detailed studies. I think I've got maybe a couple minutes here. I'll finish with some conclusions here on this study. So we've performed targeted lipidomics as well as aqueous metabolite analysis of CSF and a collection of Alzheimer's patients with cognitive impairment and healthy controls. And we see a number of very interesting changes that I think can be followed up with some animal work and maybe a larger sample set to try to get at some of the mechanisms that are related to transport in particular, and changes in diet and the response in the brain and the cognition of these patients. Finally, I've just spent a minute or two on a study that were performed on a large sample set of trying to develop biomarkers of nutrition. And the goal here is to measure blood and urine metabolites, primarily use targeted lipidomics, aqueous metabolites. For blood, we've used NMR and GCMS for urine analysis. And the 3 parts of the study was a controlled feeding study of 153 subjects, and then an additional 450 subjects where we have food frequency questionnaires and 4-day food records and 24-hour recalls, and then a larger case control study where we have outcome data on patients whose blood was taken 5 to 10 years before they developed a variety of different diseases, including cardiovascular disease, breast cancer, colon cancer, and so forth. Won't go too much into all the patients. We have a tremendous amount of demographic and clinical data on these patients. The diet study was done to mimic habitual diets. So this was not a sort of dietary stress, but actually the feeding study tried to mimic what the patient or the subject's typical diet would be, which is both, advantage in that it allowed kind of a careful study of what people would be doing normally. On the other hand, it didn't produce enormous changes in diet and response that can make it easier to identify biomarkers. So this is kind of a detail on the different visits where the blood and urine were collected and so forth. And some summary of the measurements that we made, the total number of metabolites that we were able to measure, and some of the reproducibility. And as I mentioned, we're interested in identifying biomarkers for dietary intake, and in terms of macro nutrients, carbohydrate, fat, and protein. So far, we've been reasonably successful in identifying a series of metabolites, which is indicated here by Model 1, for penis percentage of energy, same for carbohydrate. And then if you if you include personal characteristics, that's Model 2. And if you include doubly labeled water or urinary nitrogen, which is often used as biomarkers of total energy or protein, then these numbers are increased, and you can actually calculate the total amount of protein or carbohydrate. Fat still is very challenging for us. And finally, the metabolites contribute, but they are certainly aided by the doubly labeled water or the urinary nitrogen. This is just a list of the metabolites, and for the carbohydrate, they're mostly lipids, which is interesting. So I'm probably out of time here, so I'll just conclude by saying that we've performed a large targeted and global profiling on over 2,000 subjects with the goal of identifying biomarkers of dietary intake. The next steps now are to relate these biomarkers for disease risk in a variety of different diseases, including some neurological diseases. So I'll stop there and take any questions if there are any.